Ryan starting off? Good. Good. Have a good weekend? Going to do a little bit of studying, I hope? Okay, good. We'll start off today. So we're going to get today into translation, which will help you with some of those homework questions that I know a few of you have questions about. My standard set of announcements. You have until Wednesday, so if you still have problems, questions, concerns about the first exam, come and find me before Wednesday. That's the last day to adjust the points. You can still come to me after that about understanding why it was graded, but you don't get any more point adjustments after that. If you have concerns, come and find me. I have office hours today from 11 to 12, and Wednesday from 9:30 to 10:30, I think. If you can't make one of those times, send me an email. We'll schedule another time to meet. All right? Then we're done with exam 1. Exam 2 comes shortly after. It is this Friday. I've given you a study guide. I posted the Mastering Biology. I've given you some homework to make you practice translation problems. There's a review session tomorrow in this room, just like last time, 5:30 to 6:30, that your TIs are going to run for you. So that, if you can make that, is a useful thing. Remember, your TIs are always there to help you as well. And then because we have our exam on Friday means there's no recitation this week. You can go, but nobody else will be there. Up to you. If you wanted to, for example, on Thursday, schedule that time to get together with some people in your recitation section and study. That might be a good way to spend that time. If you're in Friday recitation, make friends with somebody who's in the Thursday recitation and get together with them and study. All right, use that time wisely. And then you guys can all get together on Friday and celebrate that the exam is over instead of going to recitation. All right, are there any questions over the exam before we get started today? Yes?
Were all the points updated already? Like some?
No, they haven't been. I'm sorry.
Oh, okay.
I will do that when I get back to my office. Sorry, in my brain I thought I had done that, but I actually haven't. So your points haven't been updated because I spaced out this weekend. I will do that as soon as I get back to my office. Thank you for reminding me. Any other questions? All right. Then we have been talking for the last few lectures about eukaryotic gene expression, how it's controlled, well, the process of gene expression, transcription, and then how that is controlled. And then on Friday, last week, Wednesday and Friday, we were talking about post-transcriptional processing, what happens after transcription, before we get to the process of translation. Just to remind you, the central dogma, in its most basic accepted form, right? We're describing the flow of genetic information. There's 3 processes generally represented in the most basic form. DNA replication, the DNA can make copies of itself, which we've already talked about. Moving from DNA to RNA, that step of transcription, which we've already talked about. And then moving from RNA to proteins, which is what we're getting into now, that process of translation. All right, we've added some extra bits to it. For example, DNA to RNA, mRNA to proteins. But this is the central dogma in its original form. All right, so I just want you to recognize that. The step that we are on, well, that we've been covering is that RNA to mRNA, that processing step, including capping, splicing, and polyadenylation. And you need to know each of these steps. You need to know for capping how to draw that 5' cap. with the central components we talked about, the methyl group, the 5' to 5' linkage. Splicing, you need to be able to draw that branch point with the 5' to 3' standard linkage, as well as the 5' to 2' novel linkage, which is what forms that lariat structure. And you need to be familiar with the two transesterification reactions that result in intron splicing, putting those exons back together. So this is where we're at. We've talked about transcription, how that produces a primary transcript or a pre-mRNA, what that pre-mRNA looks like, and then how that is processed into an mRNA by removing those introns, adding the cap and the polyadenylation so that you have an open reading frame within that mRNA. So our first question today, what is the structure of that primary transcript? Make sure everyone's on the same page. And understand primary transcript versus a processed mRNA.
But then just now she showed a picture and it was the primary transcript didn't have the capital. So it's yeah, she just okay, I just walked into it. But yeah, she emailed us.
All right, everyone, get a chance to read those over. Get your answer in.
Maybe one of us will get it. It's either A or E.
Let's take a look. Make sure everyone is understanding.
I don't know. It's either A or E.
All right, so there are your responses. The correct answer is A. All right, the structure of the primary transcript, the thing that is produced by the process of transcription directly, is complementary to that transcription unit in the gene, but it's before it's been processed. So it hasn't been capped, spliced, or polyadenylated yet. Once that happens, we call it an mRNA. It's no longer a primary transcript. All right, we also looked at the process. We ended up sort of very quickly with the process of the polyadenylation. There is a recognition sequence at the 3' end of a primary transcript. That AAUAAA. There's an enzyme that cleaves downstream of that. And then we have another enzyme, a poly-A polymerase, that's just going to add a series of A's to that 3' end. Remember, the function of this, like the 5' cap, is to protect the mRNA from degradation once it moves out into the cytoplasm. And it also helps with recognition for that transport process out to the cytoplasm, as well as recognition by the ribosomes that that RNA is an mRNA that's coding for a protein. All right, so that poly-A tail is very important to that mRNA and proper functioning and getting to the right place. We ended on Friday with this question. So if that's the way, the process of polyadenylation, adding that poly-A tail, what does this mean about the actual 3' end of the primary transcript? Right? And the non-poly-A end of the mRNA. How are they related? All right, so there are your choices. Think about that for a minute. Think about the process of polyadenylation, what's happening, and what that means about the 3' end of the primary transcript versus the 3' end of the mRNA. Read those choices carefully. Talk it over with your neighbors.
says you just add on.
You guys have to toss it over a little bit more before I do that.
I just thought it was like.
All right, look at each one carefully. Think about the process and what that suggests.
Wait, wouldn't it be?
You guys have it all figured out?
Are they just saying that?
Wait, no, it's not the same one. It's not.
The first part that gets transcribed.
That PDA, the poly, that's not the same.
So is it?
He's drawing it out. Wait, Lynn.
Okay, you're right.
Wait, hold up. Wait, it is present. Wait, Liv. It is present. But it's not the same one.
All right, everyone get their answers in. Give you one more quick second to decide.
I don't know. Most of the time you're right, like 3 out of 4 times. Like 9 out of 10.
A minus. A plus.
I didn't get 4.
I'm probably like 20.
Like, what is your age? The first 3. So then you can use that one because
I did that once.
I don't know what
We'll go over it. Do your best. Yeah. Sorry, what's the question?
it is present.
I mean, the complimentary.
This is okay with all these and answer. I don't know why the answer.
Wait, is it Eva?
All right, everyone get their answers in.
Wait! Lynn, switch to E. I did. That was my first letter. Wait.
All right, let's go ahead and take a look and see what you think.
No, first farmer check only happens before between process.
All right, before I show you the answer, let's go over this one by one. So we're talking about the 3' ends of the primary transcript, right after transcription, before processing. All right, so is the 3' end of the primary transcript the same as the 3' end of the mRNA? Why? Right? So the 3' end is cleaved. If we ignore that poly A tail, it's cleaved off. So it can't be the same. B, is the 3' end of the primary transcript, and by part of the transcription unit, I mean it's coded for in that transcription unit.
No.
Oh,
Cut off.
disagreement. Defend your answer. Is it yes or no?
Yes. No. Yes.
Wait, are you talking about the part that gets leaved in or the part that gets removed?
We're talking about the primary transcript
The
before it's
definition of the
processed.
primary transcript is
It's what's transcribed.
after transcription, which is from the transcription unit.
All right, so after transcription, right, we're transcribing that transcription unit. Everything in that transcription unit is transcribed. The primary transcript is a direct copy of that. Therefore, the 3' end has to be coded for in that transcription unit. Does everyone see that? All right, what about C? The 3' end of the primary transcript is not present in the mRNA. Why? Because it gets cleaved off as part of that polyadenylation process. All right, so let's try one more time. I'll give you another chance. What's the right answer then?
Was it
Yeah, Lily just asked.
All right, so we just went over which ones were correct. What's the right answer?
We were right.
Looking at now,
What, me? I'm just trying to wake up.
I think my clicker has, my remote doesn't really like Monday. I think that's just generally the problem. All right, what's the right answer? E. All right, it is part of the transcription unit, but it is not present in the mRNA.
But you're right.
All right. So that, we're going to leave processing now, and we're going to start looking at the process of translation, how we get from that mRNA sequence to the sequence of amino acids we find in proteins. So we're going to go from mRNA to protein. So we've gotten from DNA to RNA, RNA to mRNA. Now we're going to move from mRNA to protein, the process of translation. All right, we're going to start off talking about how we draw a polypeptide. All right, so what is this representative, this structure? It's a basic amino acid structure. All amino acids have that same structure. Hopefully you recognize by now because you've been learning them. All right, it's got an amino end, carboxylic end, and then some side chain that defines what amino acid it is. But they all have that same structure. And we want to turn this single amino acid into a polypeptide. All right, multiple amino acids linked by peptide bonds into a protein. So if we look at two examples of amino acids, notice that now that R group is flipped and pointing down. This is a convenient way to draw it, I find. It doesn't really matter which way you draw it, but you're going to see me draw it either way. I like doing it this way personally. But we're looking at how we're going to put these two things together. We're going to link up that carboxylic end with the amine end. What kind of reaction? Condensation reaction. So we are going from these individual amino acids to condensation reactions to form peptide bonds, the bond between these amino acids. The bond in particular is between the amine group of one amino acid and the carboxylic group of another amino acid. And the backbone is the same for all proteins. It's that repeated structure. Right? We have the amine, we have the carbon that's the side chain that R group comes off, and then we have the carboxylic end. Amine, side chain, carboxylic end. So you can start off drawing any amino acid by drawing a set of this repeated structure with the number of amino acids you have, and then just fill in the side chains. All right, it's a repeating structure. It is the same for all proteins. Where are the carbonyl groups? What is the carbonyl? Right, it's that double bonded O that's left after our condensation reaction. Where are the amines? Our NHs. All right, which means that C, that carbon between them is left for the side chain. So here are two amino acids put together. Anybody recognize those amino acids? Aspartic acid is the first one. What's the second one? Phenylalanine. Phenylalanine is nice and easy because it's got that ring structure. What's the chemical name of the compound now that we put two amino acids together?
Polypeptide.
Polypeptide. All right, so we've gone from single amino acids to two or more linked in a chain. We've gone to our polypeptide. All right, notice it's drawn charged, again, because we are in that aqueous solution in the cell. That affects those functional groups. All right, so if that's how we're drawing, right, in this class, our peptides, how do we get to that sequence from our nucleic acid sequence, from our mRNA? So we've looked closely at the anatomy of a gene, and a few of you are getting confused when I asked for the anatomy of a gene with the anatomy of a primary transcript versus the anatomy of an mRNA. So make sure you go back and look at those three things. They're distinct. They have distinct components we're concerned about. All right, the anatomy of an mRNA. It has a region that is translated, right? This is the thing that's moving out into the cell and being read and turned into a protein. What is that region that's translated called? It's an open reading frame, or ORF. All right, that region that is translated is also called the protein coding region. The open reading frame. So in our schematic here, it's that pink bit. Remember how this is different from a primary transcript, which has exon and intron structure. So that open reading frame is translated into a functional protein, a giant black arrow. But that is only the pink region of our mRNA. What happens to those blue regions? What happens to those blue regions? The question is if they are degraded. They are not. It's a good guess. But they are an important part of our mRNA. Well, that's what defines our open reading frame. What about outside of that? Are those blue areas translated?
No.
So we have an untranslated region on either side of our open reading frame that is not coding for our protein sequence, but is important for getting that mRNA hooked up into the ribosomal protein-making machinery. All right, so in our mRNA, we have that translated region buffered on either side by untranslated regions, but they still have a function. We'll look at that when we get into the mechanics of how ribosomes are actually making proteins based on these mRNA sequences, which will be, I think, after the next exam. But don't forget, when you draw an mRNA, it is not all open reading frames. There are untranslated regions on both the 5' and the 3' ends that are important to that structure. All right, we've already mentioned, you brought up, that we're defining this open reading frame by a start codon. So open reading frames, eukaryotes always start with the same amino acid, methionine, which is coded for by the triplet nucleotide sequence AUG. There's only one way to code for a methionine, so this one is an easy one to remember. This one you should recognize without needing a table. Right? Open reading frames always start with an AUG. On the other end, we know where our open reading frame stops by the presence of a stop codon. There are three possibilities for stop codons: UAA, UAG, and UGA. So if we find the beginning of our open reading frame, by the presence of that AUG, we know where that protein coding region stops by the first stop codon that we run into. After that, it's not translated, right? That's the signal to stop translation. In between, we shouldn't find any stop codons, right? The start and the stop. In between, it'll be all amino acids. Also notice, much like some of the other important biological molecules we've looked at already, proteins have a directionality that is important. At the beginning, because of the way these reactions happen and the orientation of the amino acids, we can refer to the ends of a protein by the amino terminus, so the side that has the free amine group. That's going to be represented by the 5' end of our open reading frame. All right, we can move through our open reading frame so we get to the other end. So the carboxy terminus, represented by the 3' end, the end with the stop codon, the end of the amino acid that has the free carboxylic group. All right, so we can translate. So this should start looking familiar based on some of the homework problems that I gave you for the weekend. At the beginning, if I give you a chunk of sequence and I tell you where it's located overlapping with that beginning part of the open reading frame, you're looking for an AUG to figure out where that open reading frame starts. You're looking for that methionine. All right, so if we think so about the methionines and the AUG, how many are there in a polypeptide? See if my clicker device is woken up. Maybe not so much.
Well, actually, I've only actually seen, just because I haven't written MT and maybe
All right, talk it over with your neighbors.
Is it B?
We'll do a show of hands here momentarily.
I thought it'd be a
Not bad.
It's
Let's
not
do this by show of hands. So how many think that there are 0 methionines in polypeptides? How many think that there's 1 but only at the beginning? How about 1 but only at the end? About D? Can't tell without looking at the open reading frame. Or how about E? None of the above. All right, the correct answer, get my microphone back, is D. You can't tell without looking at the open reading frame. So, a protein, a polypeptide sequence, the start is always signaled by the presence of the methionine. But the methionine is an amino acid just like any of the rest of them. So it can happen, you can find it internal in the protein. So don't get confused if I ask you to translate something and there are multiple methionines. It doesn't mean there are multiple starts. First one signals the start, the second one is just part of the amino acid chain.
So does that,
Yeah, question.
does that mean all the open reading frame starts with a methionine then? Like all the sequences?
If, right, every single mRNA that's made into a protein, that methionine signals the start. But if I give you something from the middle here, you're not going to necessarily have a methionine as the first amino acid, right? It's only here that you find, you know you'll find a methionine. Right? If I'm giving you a chunk of sequence and I indicate that it's coming from the middle of the open reading frame, start with whatever amino acid I decide to start with, really. Right? It's the middle of that open reading frame. You don't expect to see it start with a methionine, or what else don't you expect? A stop codon anywhere in there. There's only a stop codon here. There's a methionine for sure here, although it can occur other places. Here is the only place you should find a stop codon. All right, so if it's at the end, you know that you're looking for a translation of your mRNA sequence that contains the stop codon. If it doesn't have a stop codon, it's not the right translation. All right, so you have to use the information I give you as far as what part of the open reading frame I'm asking you to translate from mRNA into amino acid sequence to figure out what the right reading frame is. All right, so how is that open reading frame read? Let's take that apart. Is it 3' to 5' or 5' to 3'? It's read from the 5' end, the mRNA, to the 3' end. How many bases at a time? 3 bases at a time. All right, so I can give you, like I did for your homework, any sequence, and you should be able to translate that into the polypeptide, the sequence of amino acids that it's coding for. All right, so for this one, we have that AUG, codes for methionine, AAA, a lysine, CGA, an arginine, and then a stop codon. That's our polypeptide. For you to be able to do this, what information do I need to give you? I need to give you the translation table. I need to give you some way to decode what these 3 sets of 3 letters mean. All right, so here is an example of a translation table. It has both the nucleic acids on it as well as the amino acids. All right, so it gives you first position. So in our triplet codon, if it's AUG, we find A. The columns give you the second position, U, and then you look within that box for the third position, G, methionine. All right, so I will provide this for you on the exam so you can do the translations I'm going to ask you to do. Deal? All right. So just to show you, there are multiple ways to represent this. Here's another way. I like this one. I don't know. Something about circle makes it pretty. Start in the middle, your first position, find your second position, and then your third position. So that AC, any of the nucleotides is a threonine. Don't like that one? I won't give you that one. I'll give you this one. Just so you know, there are multiple ways to represent that information. I'll give you this one, though. It's the most common way. All right, so let's practice. So for the rest of class today and beginning of class on Wednesday, we're going to practice translations because I want to make sure that you understand what an open reading frame is and how you find it and how you determine what the right one is based on the information I've given you. All right, so I'm going to ask you to translate this sequence. I'll give you the table in a minute to do that.
I haven't even had time right now.
So you know that? Yeah, we've got to be in our.
Yeah, the sequence at the top.
I mean, it's not going to stop.
Sorry.
Oh, okay.
It's not there.
For the homework page, we should have.
I didn't expect you to write it down that quickly.
How do we draw that? We just draw like an arrow. So it was coming from over here, not like.
Which is really, really different. No, it's not all of
There's no R.
You remember the
R is tires.
It's not more.
Oh, you're looking at the.
I got it. I need to make lines. It doesn't matter if you. So then you would just like say continue. At the beginning. Right?
You don't have to show the I'm not sure if this is the case.
Did you I still can't drive.
Yeah, I got you. I have a hard time with
How's everyone doing?
I think it's because I almost forget like which part of the
All right, everyone get that figured out? Give me 1 more minute.
She said that they sort of, that we have to know these. No, yeah, so that's why she said she didn't memorize them because she thought it was Oh, really?
All right, let's take a look at it together. All right, so that's your sequence in our translation table. Here's our sequence. We're going to start. We're at the beginning of the open reading frame. We're looking for an AUG. It's right there at the beginning, conveniently. That codes for methionine. We slide down 3 base pairs. Look at the table, see what that codes for. What is it? Arginine. Slide down 3 base pairs, the pirate amino acid. Arginine. Down 3 letters, what's the next one? Glycine. Down 3 base pairs, what's the next one? Alright, so leucine. Down 3 base pairs. Tyrosine, did I skip a letter? So there was a question up front, so it should be UAC. And then what should it be? Tryptophan. That's what I get for doing it early in the morning. Alright, so this demonstrates when you do it quickly, even people like me who have done it a lot make mistakes. It's hard to keep track of your base pairs, so be careful. Alright, it's really easy to shift over 1 base pair and make a mistake. So if we do it correctly, and it's a tryptophan you said? No? It's a tyrosine, I've just missed a base pair. So that leaves 1 base pair at the end, that U. So the question is if that was intentional. So when I give you a sequence, particularly when it's at the middle, right, you have to check every possible reading frame. Which means sometimes there's going to be bases hanging off the end that aren't in sets of 3. Can you tell me what's there based on that single single nucleotide? U. No. What about if I made it a UA? Can you tell me what that next amino acid would be?
No.
Yeah, I'm sorry. No, it can't stop because she's at this part of the open reading page. So it has to be
Right, if this is in the middle, could you tell me what it was going to be? It would have to be a tyrosine if it was in the middle because we were not looking for a stop codon. Alright, so sometimes, even though you don't have a triplet codon, you can still tell what the amino acid is. Why is that? What's unique about the 3rd base pair in a triplet codon?
So that's what she just did.
For many of the codons, there are multiple ways to code for the same amino acid. Right? That's called code degeneracy. So the first two letters in the triplet codon are more important often than the third one. Oftentimes that's a wild card. So if you look at alanine, for example, doesn't matter what that third base pair is, it always codes for an alanine. So if there are two base pairs hanging off the end, you might still be able to tell me what the next amino acid is. Right? Based on the information I've given you about where it is in the sequence and what you know about how the triplet code works. So pay attention to those. All right, are there any questions about that? I have explained it to you in class. It is fair game for a question on the test. All right, don't ignore those last few base pairs hanging off. If there's one, you can't tell. If there's two, you might be able to tell. You might not, but you might be able to tell what the next one is. Is there a question, maybe? So the questions on the homework, do you assume that you always start with the first base pair?
Well, more likely than not.
Yeah, but she says in the middle.
Well, so for the homework, I've asked you many questions, and you have to figure out what the right reading frame is, whether it starts with that first nucleotide, the second nucleotide, or the third nucleotide.
How would you know if it's like
Right? When you're looking at the genetic code, you don't always start, you don't always know where that reading frame starts, so you have to find it. So the